My dear students, in this class, let us learn about the history of cell biology. Cell biology means study of life at cellular level, whereas cytology is the study of cell itself. The difference between cytology and cell biology is study of cell is called cytology and study of life at cellular level is called cell biology. Cell is the basic unit of all living organisms and cell is nothing but a compartment. So, in our body, there are various cells, and each cell acts like a compartment, and each compartment is differentiated or separated from other compartments by plasma membrane. So, cell means it's a compartment, it's a chamber. Cell biology deals with all aspects of cell, including structure and metabolism. So, cell is nothing but a compartment inside which various metabolic activities will be taking place with the help of various cell organelles. Now, Robert Hooke has identified cell as a compartment. In fact, he did not observe the living tissue, he only observed the dead tissue, that is cork. That too in 1665 he made this observation when there were no proper microscopes, when there was no technology. Robert Hooke first identified the compartment like structures in the cork in 1665. And Robert Hooke identified cork, which is nothing but a dead tissue. This is the microscope that was used by Robert Hooke, and he wrote one book. Robert Hooke wrote Micrographia, and in this Micrographia, he recorded all the characters of the cell and he made the recording of his observations. So, Robert Hooke authored one book called Micrographia. Robert Hooke observed only cork. He identified it only as a compartment. He did not observe any cell organelles. Robert Hooke identified only the dead tissue. This is exactly the compartments identified by Robert Hooke. So, Robert Hooke identified compartment like structures in the dead tissue called cork. Later, Antony van Leeuwenhoek came into picture, and Antony van Leeuwenhoek discovered protozoans, and he called them as wretched animalcules because Antony van Leeuwenhoek did not know the significance of protozoans. And after all these discoveries, with the passage of time, scientists came to know that protozoans cause many diseases. And the protozoans may disturb our economy negatively and also may boost up our economy positively. They have got their own significance, but Antony van Leeuwenhoek did not know during the time of the discovery of protozoans all these facts. He used simple lenses and he observed certain moving specks, and he named these moving dots as protozoans. So, Antony van Leeuwenhoek discovered protozoans and called them as wretched animalcules. And the same Antony van Leeuwenhoek observed spermatozoa and also erythrocytes. And these are the various instruments used by Antony van Leeuwenhoek. He was properly regarded as Antony van Leeuwenhoek was properly regarded as father of microbiology. And he not only observed the animalcules, Antony van Leeuwenhoek not only observed the protozoans, he also observed the various parts of the multicellular organisms, various parts in the vertebrates, invertebrates. And these are the spermatozoa observed by Antony van Leeuwenhoek in age-old days. Cell theory was proposed by G.L.N and Schwann. Rudolf Virchow stated that cell arises from pre-existing cell. The cell theory states that this is old cell theory. The old cell theory or the traditional cell theory states that all living things or organisms are made up of cells and their products. New cells are created by old cells dividing into two. Cells are the basic building units of life. It's a well-known fact that cells always exhibit cell division. Prokaryotes exhibit amitosis, whereas eukaryotes divide either by mitosis or by meiosis. And there are certain exemptions to cell theory. Viruses are not considered as cells because virus, as long as it is present outside the body, it is comparable to a chemical crystal. And first formed cell during the process of evolution, they may be conservates or protobionts, they are exempted from this cell theory. And mitochondria, chloroplast, which are nothing but semi autonomous structures within the cell, are again some exemptions to cell theory. Now, let us come to the modern cell theory and its various principles. All known living things are made up of one or more cells. All living cells arise from pre existing cells by division. The cell is the fundamental unit of structure and function in all living organisms. The activity of an organism depends on the total activity of independent cells. Energy flow occurs within cells. We know that cell contains mitochondria. Mitochondria is a powerhouse of cell which generates ATP. And unless there is energy flow, you can't call a cell as living. So, in order to sustain the cell, this mitochondria has to generate ATP. Cells contain DNA, which is found specifically in the chromosome, that means within the nucleus, and RNA is also found in the nucleus along with the cytoplasm. DNA is found only in the nucleus, whereas RNA is found in the nucleus and also in the cytoplasm. All cells are basically the same in chemical composition in organisms of similar species. That means the body of the members belonging to a same species is made up of similar types of cells. That means there is no chemical variation, there is no physical variation within the members of a species. Purkinje named the cell contents as cytoplasm. Purkinje is the scientist who coined the term cytoplasm. Waldeyer used hematoxylin for staining the tissue cells. That means this technique of staining the cell was also developed by this Waldeyer, and this Waldeyer used hematoxylin for staining the tissue cells. His is the name of the scientist. His is the scientist who developed microtome for cutting sections of tissues for cell study. We need certain transverse sections, longitudinal sections for our observations. We'll be doing this section cutting in our labs. And for this, one scientist by name His developed one instrument called microtome. And microtome is the instrument developed by one scientist by name His for cutting sections of tissues useful for cell study. So, Van Benden discovered centriole. We know pretty well that centriole is the structure that is responsible for the generation of spindle fibers. And spindle fibers pull the chromosomes to the poles during cell division. Unless there are spindle fibers, cell division is not possible. And spindle fibers are generated by centriole. So, cells, in order to exhibit cell division, centrioles should be present. And the neurons, they don't contain centrioles. As a result, neurons do not exhibit cell division. And here you can see one more scientist name. Mechinkov observed and named phagocytosis in the cells. Phagocytosis is nothing but cell eating. Devouring of solid particle is called phagocytosis. And this term phagocytosis was coined by Mechinkov, and he has also observed this phagocytosis for the first time. Louis Pasteur observed the anaerobic release of energy from yeast and molds. That means this fermentation process was observed for the first time by Louis Pasteur. Apart from this, there are various cell components observed by various scientists. Porter observed endoplasmic reticulum. Camillo Golgi observed Golgi complex, like that, which we already discussed in our previous class. So, this is about the chronological order of cytology and cell biology. Thank you very much.